Residual solvents are one of those hidden details in pharmaceutical manufacturing that most people overlook until something goes wrong. Today, we're going to walk through a complete, practical guide to Class 1, 2, and 3 residual solvents, why they matter, and how the US pharmaceutical industry controls them. These solvents are leftovers from manufacturing steps like synthesis, crystallization, purification, or drying. And even though they're invisible to the naked eye, they can impact patient safety if not properly monitored. Let's dive into the framework, the testing processes, and the strategies that keep our medicines safe. Residual solvents are not impurities formed during degradation. They're process-related chemicals that remain after manufacturing. To understand why these solvents matter, consider this real scenario. A small pharmaceutical company rushed a generic product to market. Everything looked fine. But during routine QC testing, a small peak popped up on the chromatogram. It turned out to be benzene, a dangerous Class 1 solvent, carried through from a supplier's raw material. That single peak led to destroyed batches, months of investigation, and severe financial loss. This is why residual solvent testing isn't just a requirement, it's a safety barrier protecting both patients and manufacturers. In the US, residual solvent testing is governed by USP 467, which is aligned globally with the ICH Q3C guideline. These regulations categorize solvents based on toxicity. Think of the classification system like a traffic light. Class 1 is red, solvents that are never acceptable with high toxicity. Class 2 is yellow, solvents that pose moderate risk but are often necessary in production. Class 3 is green, solvents with low toxicity and minimal concern. But here's the catch. These classifications only matter if you actually test and confirm compliance. Without testing, you're flying blind. Class 1 solvents are the highest risk category. These are human carcinogens and should be avoided entirely in pharmaceutical manufacturing. Their limits are extremely strict, often in the low parts per million range and sometimes even lower. Common Class 1 solvents include benzene, carbon tetrachloride, dichloroethane and trichloroethane. And here's a key point. Even if you never use Class 1 solvents intentionally, contamination can still sneak in through recycled solvents, raw materials, or upstream suppliers. That makes monitoring absolutely essential. Next, we have Class 2 solvents. These are substances that pose toxicity risks but are still widely used because they're effective in chemical synthesis and process operations. Examples include methanol, toluene, acetonitrile, chloroform, and THF. The limits for Class 2 solvents vary based on toxicity. Some can be present at thousands of parts per million, while others must stay tightly controlled. A major challenge with Class 2 solvents is batch-to-batch variability. Different manufacturing steps can contribute different residual levels, so mapping solvent usage and tracking variability is essential for compliance. Class 3 solvents are the least risky, the green light solvents. They have low toxicity and are generally allowed up to 5,000 parts per million without special justification. Common examples include ethanol, acetone, isopropanol, and heptane. But even though they're low risk, they shouldn't be ignored. Excess levels can still affect product stability, odour, and impurity profiles. A best practice is to set internal limits lower than the pharmacopeal limits to ensure consistent product quality USP 467 uses a tiered testing approach based on headspace gas chromatography. Here's how it works. Procedure A is the screening test. If no solvents exceed limits, you're done. If solvents are detected, you move to Procedure B, which uses a second column to confirm their identity. If Class 1 solvents are suspected, Procedure C provides targeted testing. And for specific Class 2 solvents, Procedure D is used for quantification. Many labs create faster, validated in-house methods, especially when monitoring multiple solvents, but they must still be scientifically justified. Residual solvent testing isn't just about following USP, it's about running an efficient and reliable laboratory. Sample preparation is critical. Using the wrong diluent or poor vial sealing can cause solvent loss or inconsistent results. Headspace conditions, like equilibration time and temperature, directly influence volatilization and recovery. For calibration, external standards often work well, but for highly volatile solvents, internal standards provide stability. Column choice also matters. USP recommends G43 and G16, but many labs prefer low-bleed modern columns for better reproducibility. And one of the most common challenges is coelution, when two solvents overlap on the chromatogram. That's why USP uses two different columns in Procedures A and B. Residual solvent control is more than just testing. It's a complete quality system. A strong control program includes supplier qualification, material traceability, routine impurity profiling, method revalidation after process changes, and trending of historical solvent data to catch issues early. Regulators expect a clear, scientific justification for your chosen analytical method, especially if you're modifying or replacing USP 467. These systems help prevent failures like the benzene incident we discussed earlier, and they ensure both patient safety and regulatory confidence. Residual solvent analysis is ultimately a protection system. It protects patients, protects product quality, protects companies from costly recalls, and protects scientific integrity. To recap, Class 1 solvents are carcinogenic and must be avoided. Class 2 solvents are toxic but sometimes necessary, requiring careful monitoring. Class 3 solvents are low risk but still need to be controlled. USP 467 gives us a structured framework, but it's strong analytical discipline and continuous oversight that truly ensure compliance. By understanding the differences among these solvent classes and applying consistent testing practices, we keep medicines safe, effective, and trusted.